As far as Chapter 10 is concerned, we're just going to look at overall what's going on with regulatory strategies. So just an overview of these strategies. Not a whole lot of detail here. And what you should be able to do by the end of this chapter is list, define, and provide an example of each of the different types of regulatory strategies. So there are 5 principal regulatory strategies. These strategies include allosteric control, multiple forms of enzymes, reversible covalent modification, proteolytic activation, and then controlling the amount of enzyme present. So first, looking at allosteric control. These proteins will contain multiple functional sites. So we have multiple functional sites that are present. These are also going to show cooperativity. Okay, so these are enzymes. These are proteins that are enzymes, and they're showing this cooperativity, and we've seen cooperativity when it has applied to hemoglobin. So we know that what happens here is that something will bind at one site, and that then will alter the binding at another site. So in the case of hemoglobin, once oxygen bound at one site, it made it easier for it to bind at other sites. Now, the activity can be modified in response to signal molecules or to information that's shared among different active sites. So things bind, they alter the binding at the additional active sites. Now, an example of allosteric control would be the enzyme, change colors here. So an example is AT case. And this is aspartate, transcarbamylase, okay, otherwise just referred to as AT case. The next example is multiple forms of substances present. So these things will form isozymes. Multiple forms are isozymes. These enzymes provide an avenue for varying regulation of the same reaction to meet specific physiological, so we're looking at physiological needs. And when we do this, we're looking at specific tissues at particular times or stages in development. So here, multiple forms are present, and the different forms that are present may have slightly different functions. And this is based on the idea that different tissues have different needs at different times. So these enzymes are expressed in a distinct tissue or organelle. So the expression occurs in a distinct tissue or organelle at a distinct stage in development. And an example of isozymes would be something like lactate dehydrogenase. So depending on where you are in development, you might have a different form of this present to meet the needs at that time by that tissue. Next, we have reversible covalent modification. So here what we do is we alter the enzyme by covalent attachment. So the catalytic properties, the ability of that enzyme to function, or how fast that enzyme is turning over substrate molecules, this will be altered by covalently attaching some group, some chemical modifying group. More commonly than not, that group that we add to it is a phosphoryl group, and the source will come from ATP. Although there are other substances that can serve as these covalent modifying groups, but the phosphoryl group is more common. ATP is what's going to give us that phosphoryl group in a lot of cases. So remember our catalytic properties, we're looking at the rate at which that enzyme is going to function. So adding a phosphoryl group or removing it is either going to speed it up or slow it down. An example of reversible covalent modification are protein kinases. Next, we have proteolytic activation. And what happens here, we have the enzyme cycling between active and inactive states. So we have forms, we have a form of the enzyme that is active and will actually act on whatever substrate it's supposed to, and then a form that is inactive. Many of these inactive are activated by hydrolysis of some peptide bond somewhere in the overall structure. The inactive precursors to these active enzymes are referred to as zymogens or proenzymes. So that's the inactive precursor here. Now, examples of the proteolytic activation: digestive enzymes are enzymes that tend to be present in an inactive form until we need them, and then some enzymes that are associated with the blood clotting process. So an example of these digestive enzymes that we've looked at, something like chymotrypsin. There's an inactive form which is then activated. And this really would be advantageous, because if you had the digestive enzymes present in the active form all the time, it's going to start working on tissues that you don't want to digest. So you only want it present in response to certain physiological needs, and then it becomes active. And then the last type of regulation is simply controlling the amount of enzyme that we have present. Now, this is going to occur at the level of transcription. So when transcription occurs, that's the place where we'll be able to control how much of that enzyme is present. So enzyme activity can be regulated if we adjust the amount of enzyme that we have there. So we don't need as much activity, we don't need as much enzyme present. So these are the different types of regulatory strategies. Again, you should be able to list and define these and provide an example of where you might see this type of regulation. And a lot of the enzymes that we're looking at here, we will explore in more detail, or have explored in more detail, in other chapters and specific contexts.